Hello, everyone. I just want to welcome you to the next installment of Vibrant's educational webinar series. Today, we are, again, incredibly excited and honored to have Dr. Tom O'Brien here as a guest speaker for us, and he's going to be discussing some topics that relate to the mechanisms of autoimmune disorders. I am your host, Sarah Ashman. I'm a clinical dietitian and marketing liaison at Vibrant. And Dr. O'Brien, if you're not familiar with him, I'm sure there's not very many people who aren't at this point, but in case you're not familiar with Dr. O'Brien, he is well known in the functional and integrative medicine world for his work in terms of gluten and wheat sensitivity, intestinal permeability, as well as autoimmune disease. He's a recognized and sought after speaker, workshop leader. He does quite a lot of work. His own program, the glutensummit.com, is actually something that you guys should definitely check out if you haven't ever registered and listened to that. It's a fantastic online summit. Basically, Dr. O'Brien has done so much to advance, the eye on gluten and wheat in the American food supply, and maybe some of the implications that it has for human health, chronic disease, as well as underlying autoimmunity. His book that he wrote in 2016, The Autoimmune Fix, won the National Book Award. It is still, I think, one of the top sellers on Amazon, I believe, even a couple years later. So, Dr. O'Brien, without any further ado, I'm going to turn it over to you and let you go ahead and jump in and tell us all about autoimmune disease and mechanisms underlying it. And once again, thanks for being here with us today.
Thank you, Sarah. I'm going to activate my slides. And Sarah, can you tell me if my slides are on your screen now?
They are not yet.
Okay.
There might be just a little bit of a delay.
Okay.
We'll give it just a second.
Maybe I need to hit share screen. Let's see.
Yes, you do.
Okay.
Yep. Hit share screen.
Share screen there, and it says this will stop other screen. Do you want to continue? Yes. Sorry, Sarah. I'm going to bump you.
It's okay. This is a tutorial on our webinar software. That's okay. All right, there you go. I can see your slides, and if you want to go ahead and just go into full screen, that way everyone can. Perfect. All right.
There we go. All right. Thank you. Thank you.
You're welcome.
I would start by addressing this photo, and that I use this concept quite often now, actually all the time, in explaining functional medicine to people. And please feel free to use it or any version of it, modified any way you like. But Mrs. Patient, when a person comes in with symptoms from, diabetes or migraines or rheumatoid arthritis or recurrent miscarriages, whenever they come in with symptoms, it's like they've fallen over a waterfall and crashed into the pond below. And they swim up to the surface and they spit out the water. They're trying to just stay afloat. And they're trying to stay afloat in this pond of diabetes. But it's very hard to stay afloat because the water is so turbulent. The waterfall keeps falling into the pond. And so they're trying to stay afloat. And everyone's looking for the life jacket to stay afloat in the pond of recurrent migraines or recurrent miscarriages or diabetes or rheumatoid or psoriasis. And it's really important to get a life jacket to stay afloat in that pond, in that environment you're in. However, and you always want the life jacket with the least side effects possible. However, once you've got a life jacket on, you have to swim over to the side of the pond, get out of the water, walk up the hill, and walk back up the river and figure out what the heck fell in the river that carried me downstream, and eventually I crashed into this pond of diabetes. And patients get that. They understand that visual. So please think about using that analogy or one like it of your own, whatever you like, to give them the big picture vision that although we want to help them stay afloat in their symptoms, we need to figure out what the heck happened here. I just came back yesterday from the 11th Annual International Congress on Autoimmunity in Lisbon, Portugal. And it was a remarkable experience to hear some of the cutting edge things scientists are talking about, and I'll tell you some of them today. But we now have a world called autoimmunology. It's a whole new discipline within the field of immunology. And there's talk of board certification within the next 3-4 years in autoimmunology because it's growing so quickly. As you may know, autoimmune diseases are a disease of the modern era. They were quite rare in the past. On occasion, they occurred, but the exponential rise in autoimmune diseases is because, bottom line, because of the way we live our lives now and the environmental exposures that we're always in touch with or exposed to. So premise number one, when is it normal and necessary to produce antibodies to self? Why are there antibodies to self? When is that normal? And no one can say it better, I think, than Dr. Terry Walls. So I'm just going to give you this.
Autoimmunity is a process where the immune cells begin attacking self and attacking normal, healthy, vibrant tissue, causing damage. And over time, we've been converting many of our chronic diseases into possibly immune, probably autoimmune, and then definitely autoimmune in nature. I would predict that another 20, 30 years, the vast majority of our chronic diseases will have been reclassified as probably autoimmune in nature.
When is it normal for the immune system to be attacking self?
So the immune system, our immune cells, have a very important function. They're involved in maintaining and repairing the body. So if my overtrain and I've damaged my muscles from overuse, my immune cells have to come in and dissolve that damage, mop it up, and repair. If I have trauma, my immune cells have to come in and repair that trauma. Or if I'm damaged from infection, they will again come in, mop up the damage, and repair. So they're a vital part of maintenance and repair of everyday and acute damage. When it becomes pathologic is when they're attacking healthy tissue, or when during the repair phase that attack continues once the repair has been complete.
That's a brilliant overview of the function of the immune system, and none of our guests have talked about the repair phase, and that explains why sometimes you have to be able to get rid of the old damaged cells. And
it's vital. We need to do that when there's damage from infection, from trauma, from overuse,
or even exercise, like a bicep.
Absolutely. And when we strength train, we are intentionally damaging our muscles just a bit. So my immune cells have to come in, repair the damage, and rebuild a stronger muscle cell, a stronger tendon, a stronger bone. And so it's a vital part of our ongoing healthy vitality.
So it's a good thing for the immune system to be getting rid of our old cells, our damaged cells, but then it gets to be threatening when the immune system is getting rid of more cells than it should be.
Correct. And when it's getting rid of cells that are otherwise healthy and vital, when, for example, I've developed an immune response to an infection, we've cleared out that infection, that clone should be wiped out and eliminated.
So I think that is a vital concept to understand. I used to wonder, why is there a normal reference range for thyroid antibodies, for TPO antibodies, for myelin antibodies, for bone antibodies? When is it normal to have antibodies to your own tissue? And it's just a normal process of clearing up old and damaged cells to make room for new cellular growth. And if you explain that to patients so that they know that antibodies aren't bad for you, excessive antibodies are bad for you, then it helps for them to ask the question, well, why do I have excessive antibodies? And that's what this talk is about. And so let's just move on. What triggers the activation of the autoimmune mechanism? The influence of the environment on the risk of developing autoimmune diseases is paramount. Paramount. In fact, more than genetics, environment shapes immune system function. Studies have shown that genetic predisposition accounts for about 30% of all autoimmune diseases, and 70% is toxic chemicals, dietary components, gut dysbiosis, and infections. And so this is the drawing that is really great to have. And in your practices, you may want to print out this in color and put it in a clear plastic folder in your treatment rooms to show to people and say, you're in the center here, Mrs. Patient, in this autoimmune mechanism, or it's progressed all the way to an autoimmune disease now. You've killed off so much tissue, it's an autoimmune disease. But these are the factors that we have to look at. And oh, by the way, I'll just tell you now, I'm giving you all the studies, all 56 studies that I'm going to reference here. I'm giving you all of them, and I'll tell you how to get them in a little bit. So environmental factors, including drugs, vaccines, infectious agents, contain substances with the characteristics and activity of adjuvants. Adjuvants mean it stimulates, it comes from the Latin adjuvare, meaning to turn on, to activate. They're used, adjuvants are used in vaccines, but even when they're not used in vaccines, things that are piggybacking, if you will, an adjuvant piggybacks on a vaccine. That's what mercury does. That's what aluminum does, is it activates the immune system to whatever the viral or bacterial component that we're trying to develop immunity to. It piggybacks in that vaccination. Inclusion of the autoimmune ecology, the environment, in disease etiology and health will improve the way personalized medicine is currently conceived and applied. So this is a big picture concept, folks, that I think it's really important to have and that you give to your patients. When they come in with rheumatoid, you don't just give them things to reduce their inflammatory symptoms. We have to get them to understand the ecology of where this thing is coming from, or whatever great anti-inflammatory, anti-pain, anti-skin irritation, psoriasis, vitiligo, whatever your protocols are, they're going to be short-term if we don't deal with the ecology, the environment where this stuff comes from. And these are some of the more common ones. There's, of course, the emotional, the socioeconomic status, drugs and vaccines, tobacco, even secondhand smoke is just as bad. Chemical agents, alcohol, coffee, other foods, infectious agents, they all are part of the ecology of the autoimmune cascade that sets it off. Chronic autoimmune diseases are the byproduct of the immune system recognizing self as foreign, which can lead to inflammation and destruction of specific tissues and organs. So the question is, why does the body do that? A variety of mechanisms have been suggested as the means by which infections can initiate or exacerbate autoimmune diseases. One mechanism is molecular mimicry. And that's where a foreign antigen shares sequence or structural similarities, meaning amino acid structures, with self-antigens. Molecular mimicry is a prevailing hypothesis as to how viral antigens initiate and maintain autoimmune response, which leads to specific tissue damage. Immune cross-reactivity occurs when an adaptive immune response to one antigen results in reactivity to structurally related antigens. By the way, cross-reactivity and molecular mimicry are very, very similar, if not identical, in their descriptions. Cross-reactivities are particularly important in food allergies, where they often are responsible for the disorder affecting the scope of the disease, the reliability of the diagnostic testing, and also have implications for current and potential therapies. So we're going to fly now. Here we go. Initiators of molecular mimicry. First, we'll talk about viruses. Several plant viruses contain nucleotide sequences significantly homologous, meaning very similar, to human sequences. In several cases, the plant viruses contain nucleotide sequences which exactly match the seed sequence of human RNA in either parallel or anti-parallel directions. I will show you some examples. Several plant viruses contain nucleotide sequences significantly homologous to humans. The plant viruses contain, which exactly match the seed sequence of human RNA. Here's a couple of examples. References, and look at the journal. This is from the Journal of Alzheimer's Research. And I'm going to talk to you a little bit about bacteria initiating the Alzheimer's cascade, viruses initiating the Alzheimer's cascade. There's over, at my last count, 146 studies on herpes simplex 1 and Alzheimer's, that when they do post-mortem autopsy of the plaque in Alzheimer patient brains, it's loaded with IgM antibodies to herpes simplex 1, just loaded. And we'll talk a little more about the mechanism behind that. Bacteria, what about bacteria? Well, you know, the first paper, actually, I just saw Professor Ebringer. It was so cool. It was so cool. I remember 1978 in my genetics class, and the instructor came in one day, and he was really excited. He had this paper in his hand. He said, this graduate student has just published this paper from King's College in London about the correlation between HLA-B27 and a bacteria called Klebsiella pneumoniae and developing ankylosing spondylitis. And it was the first case of molecular mimicry I ever heard. This was in my genetics class. And it was Professor Alan Ebringer, who is an immunologist and a rheumatologist, board certified, who published that paper. His entire career has been based on this. Amino acid sequences have been identified with HLA-B27, this is our own genetic tissue, and 2 enzymes in Klebsiella pneumoniae. And I put them in red so you can see the similarities there. And patients with ankylosing spondylitis have been shown to possess elevated antibodies to all 3 of these peptides. When you make antibodies to these peptides of Klebsiella, if you carry the gene B27, HLA-B27, those antibodies can go after your own genes. And here is the sequence with HLA-B27, the similarities that you see. The top row is from the nitrogenases and pullinases of Klebsiella, and that bottom row is from HLA-B27 and from collagens on our own joint surfaces. So you can see how if you make antibodies to the bacteria Klebsiella, if your immune system says, we got a problem here, there's an invasion coming in, we need to deal with this, how it can get mistaken and attack your own tissue because it looks so similar. And also they looked at, and this is Professor Ebringer again, looking at rheumatoid arthritis and Proteus mirabilis. And what did they find in that situation? That the enzymes in Proteus are both virulence factors contributing to the pathogenesis of microorganism, and it's been shown to have a potent cytotoxic agent against human kidney cells, the tubular epithelial cells, and also against joint surfaces. And here are some of the correlations between the peptides in Proteus and rheumatoid arthritis patients when it goes after, when these antibodies to these peptides go after the collagen surfaces of rheumatoid patients' joints. And HLA-DR4 is the gene that's associated with Proteus and rheumatoid arthritis patients. So this summarizes the Proteus and Klebsiella with rheumatoid and ankylosing spondylitis. A similar situation is known to occur in rheumatic fever, where streptococcal antibodies were reported to be cytotoxic for heart and fibroblast cell lines because of molecular similarity between the strep M protein and the human cardiac myosin. This is why when you go to a dentist, they give you antibiotics, because when they're working in your mouth and you rinse your mouth out, most people will spit the water out into the dental basin there, and it'll be pink water or actually blood. Well, he's just created leaky gums, you know, leaky gut. Now you got leaky gums. And there are thousands and thousands of bacteria in your mouth all the time. And if some of that bacteria gets through leaky gums into the bloodstream, the immune system makes antibodies to the strep. We've all got strep in our mouths. If the immune system makes antibodies to strep that has invaded through leaky gums, the molecular mimicry with your heart valves dictates that the dentist, because it's so common a result, that the dentist gives antibiotics to prevent that. That's why dentists give you antibiotics when you have dental work done. The association of anti-cardiac antibodies with acute rheumatic fever is well documented. Prolonged persistence of strep group A antibodies is a characteristic in patients with rheumatic valvular disease. Rheumatoid ankylosing spondylitis have cytotoxic antibodies that contribute to the development of autoimmunity by activating either complement cascade, and then here come the antibodies, or natural killer cells, leading to tissue damage. In patients with ankylosing spondylitis, Klebsiella were found to be associated with a higher degree of gut inflammation, and the source of these bacterial antibodies were shown to be the jejunal region of the gut. I put this in here because we're going to talk about the gut with autoimmunity and how critical it is that we not address just the symptoms with a life jacket because they're floating and sinking in the pond of their autoimmune disease, but we have to address the gut because it's fueling the fire in so many of these people. Degradation products of bacterial cell walls and nucleic acids are found in the joints of patients with rheumatoid arthritis. And for the chiropractors out there, I thought this was a really important concept to bring to light about bacterial DNA and bacterial cell wall constituents in the joints of patients with different arthritises. Look at the percentages of patients with bacterial peptides in their joints. With rheumatoid, it was 62%. Septic arthritis, every one of them. Inflammatory osteoarthritis, when your joint looks red, 80% of them. Crystal-induced arthropathies, joint trauma, 100% of them. So the total for overall arthritis, 79% of the patients they looked at with different arthritis had antibodies to their own, to bacteria that was in their joints. The term in 1997, pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections was coined, PANDAS. And this is just a nasty condition of kids with sudden onset of motor tics, obsessive compulsive disorders, and also Sydenham's chorea is a neurological manifestation of rheumatic fever. We're still in the bacteria here, everyone. I'm just showing you some more manifestations of it that you'll see in your practice quite often once you're keeping an eye open for it. Molecular mimicry is the proposed mechanism for PANDAS and for Sydenham's. It's group A beta hemolytic strep, and because it can cross the blood-brain barrier. That's why it's so important, the neural zoomer test, which looks at evidence of a breach in the blood-brain barrier. And there will be a new updated neural zoomer coming out in the months to come, much more comprehensive. But the one we've got now is a great test to look to see, is there a breach of the blood-brain barrier allowing these bacteria to get in, triggering what might be PANDAS, what might be Sydenham's chorea, or other manifestations. So this is from that study about PANDAS and a milkmaid's grip. If you see kids or adults that have a milkmaid grip, which is kind of like, if you go on Google, just type in milkmaid grip, you'll see some videos of it. That's better than me trying to describe it for you. Picture's worth 1,000 words on that. And the mechanism of that is both antitubulin antibodies from molecular mimicry with group A beta hemolytic strep, or lysoganglioside antibodies from molecular mimicry with group A beta hemolytic stress. So here's where I'm telling you about the studies. We're going to give you all the studies. You can have them all. They'll be ready by the end of the day today. Just go to thedoctor.com, thedr.com/AI mechanisms. The title of this talk is Autoimmune Mechanisms. So just go there and you'll get all of the studies. Now, if they were free to me, they're free to you. If they were not free, I can't give them to you free. That's illegal. But I've got the links for you to the abstracts. So there you go. Hopefully, I'm going to stimulate you to where you're going to want to read more about this. And in 6 months, just reading a little bit every week, you've got this down and you can speak to anyone about it. Why do autoimmune diseases rarely occur alone and rather occur concurrently? This is why Celiac rarely occurs alone. Rheumatoid rarely occurs alone. If you do predictive autoimmunity panels, you rarely only find one self-antigen that's elevated. You usually find more than one, and sometimes a number of them. But this is the primary reason why. And this is a great article to read when you get this article. Amy Prohl and Dr. Marshall, Trevor Marshall. I was just with Dr. Marshall in Lisbon. He was talking about this subject and how the comorbidities of different autoimmune conditions, where they come from. Persistent pathogens directly alter gene transcription, translation, and DNA repair processes. Okay, once you've done a little reading on bacteriology, bacterial growth and colonization, this makes perfect sense, of course. The catastrophic failure of the human metabolism observed in autoimmune disease results from a common underlying pathogenesis. The successive accumulation of pathogens into the microbiome over time, and the ability of such pathogens to dysregulate gene transcription, translation, and human metabolic processes. And do understand, we're not just talking about the microbiome in the gut. The microbiome is through your entire body. A woman with breast cancer has a very different microbiome in the breast that has cancer than the other breast. That their bacteria are throughout our muscles, our organs, our brain tissue, that there's no area that I know of that doesn't have bacteria as a component of the environment. Hundreds of protein interactions define a chronic disease, not a defect in any single pathway. When we were learning about these different conditions, Professor Ebringer's article in 1978 about ankylosing spondylitis and HLA-B27 and Klebsiella pneumoniae, I thought that's the pathway in development of almost all ankylosing spondylitis. It's not. It's a very common mechanism, but there are multiple pathways because there's hundreds of protein interactions that occur with every chronic disease, not just a single pathway. You guys kind of wrestle with this idea. You just kind of look at it and look at the diagram that's part of that article. Just pick any disease and you will find the studies. Each spoke of this wheel represents a published study appearing in Medline, which shows a significant statistical relationship between one disease and the other. How do you explain MS and myasthenia gravis, or MS and thyroiditis, or MS and lupus, or MS and hypertension? How do you explain these? There are multiple pathways when there's dysbiosis occurring. That's why, as you read more and more about this, you focus more and more on the gut as a primary component of your treatment protocols, irrespective of what the patient's presenting with. I know that's a bold statement, but when you read the studies, it just becomes an OMG again and again and again. This picture, this drawing, this graph that was in the study, I printed it out in color and I taped it to my ceiling. And I went to, in my bedroom, when I went to bed every night, I just look at it, I go, wow, wow. And then I go to sleep, and I'd get some ideas that come, I'd wake up and I'd write them down. And, but, however you do it, but when you think about this, and then you think about, oh yeah, I've got great techniques for rheumatoid. I give curcumin in high doses, and I get them moving, and I do proteolytic enzymes, and it helps a great deal. Well, that's great. Yeah, that's great. Those are things that can help, but there are so many pathways that have to be addressed, and the way to get into affecting systemically multiple pathways is by balancing the gut. And this is the genesis of comorbidities. Immunosuppressive therapies offer short-term palliation, but immunostimulative therapies improve long-term patient prognosis. Could they say it any clearer for us? So here's a case study that I've shown before in some talks, and you may have seen this. This is a 43-year-old guy with an 8-month history of patchy hair loss. Look at the title, Cure of Alopecia areata. That's an autoimmune disease. Cure after eradication of Helicobacter. So here is his alopecia, and I put these arrows in here because most clinicians don't look at the stubble to diagnose alopecia, but that's alopecia. Just like on the scalp, that's alopecia. There's no hair growing where there should be hair growing. And he went to a dermatologist. He's got the standard protocols, and they also put him on psychiatric support, but nothing worked. Someone noticed that he had dyspepsia, and they checked him, and he had an H. pylori infection. Look at his IgG antibodies to H. pylori. They're over 4-fold above normal. So he was given the standard protocol for H. pylori, and he was told, stop all the medications for alopecia. They're not working anyway. Just stop them. And H. pylori was eradicated in 6 weeks. They confirmed it. By the way, you always go back to confirm that your protocols have worked. That's a critical component. We'll have a whole webinar on that, on why do you have to retest and how to educate patients on how to do that. And here he is 4 weeks later, and you can see the growth is coming in. Here he is 8 weeks later, 16 weeks later, and 44 weeks later. Just by killing a bacterial infection, getting rid of the antibodies to the bacteria, because there was no longer a need for Helicobacter antibodies, the mechanism to produce them was turned off. So the cross-reactivity with his hair follicles turned off also. Now, this is all about H. pylori and how common it is. I'm not going to take the time to show you guys all of these different conditions, but read this article and you'll see it's idiopathic thrombocytopenic purpura, Hashimoto's, Alzheimer's, sclerosis, atherosclerosis, pancreatitis. All of these can be because of Helicobacter. Thyroid, psoriasis, celiac disease may be because of Helicobacter. So when your celiac patients don't get well, it may be because they have a Helicobacter infection. 50% of the world's population is said to have Helicobacter. If that's true, how many people in your practice have a Helicobacter infection that you haven't thought to check for? Up to 90% in adulthood may be carrying Helicobacter. This is worldwide, by the way, up to 90%. So let's go a little more into bacterial exposures and where are they coming from. Look at the title of this one. Opportunistic pathogens enriched in shower heads, biofilms. So showerhead biofilms can have 100-fold more human pathogens than the background water that came from the pipes up to the showerhead. And this is what it looks like, bacterial colonies on inside a showerhead. Attempted cleaning with bleach results in a 3-fold increase in the load of mycobacteria. Although anecdotal, this observation is interesting in light of the general resistance of mycobacteria to chlorine, which also may be one reason for the mycobacterial enrichment in municipal systems compared to well water. There's more mycobacteria in municipal water systems than in well water systems. Many species of biofilm-forming mycobacteria are chlorine resistant and potentially can be enriched by chlorine disinfection protocols used by many municipalities. So I just threw this in here so you guys say, oh my gosh. So anytime you have a patient that's got recurrent oral pharynx, sinus, lung congestion, bronchitis, asthma, on your checklist, you have to include bacterial exposures from a shower head. You've got to rule that out. So back to more initiators of molecular mimicry. We've talked about bacteria. Let's talk about foods. Neuromyelitis optica, 75% of those patients, it's associated with IgG1 antibodies to aquaporin 4. Proteins in nature have a remarkable similarity in sequence and structure to human aquaporin 4, which is a component of your nerve cells and your blood-brain barrier. Aquaporin 4 is a component of your blood-brain barrier. I'm hoping it'll be on the updated NeuroZoomer panel when it comes out. I'm working hard to get that one in there, and I'll show you why. We compared human aquaporin 4 to plant and bacterial proteins to investigate the occurrence of it. What did they find? Spinach constitutes 20% of the integral membrane proteins. Two thermally stable members of the aquaporin family, 20% of the membranes of spinach. High similarity to a known epitope of neuromyelitis, aquaporin 4 was observed for corn and soy. And here's the graph from the Journal of Neuroimmunology. If you look at soy, corn, spinach, and tomato for S100B, myelin basic protein, those two are on the neural zoomer panel, by the way. Anytime those come back positive, you have to check the patient for a sensitivity to soy, corn, spinach, and tomato. Antibodies against aquaporin 4. They can cross-react with human aquaporin 4. They can trigger, I call it B4. My new book comes out in a few months. It's called You Can Fix Your Brain. And I'm coining a term, capital B, number 4, B4, a breach of the blood-brain barrier. Because of this kind of stuff, it's so very common. What disease or dysfunction of the brain does not require a breach of the blood-brain barrier? That's a $1 million question. Every brain dysfunction is a dysfunction, as far as I know. Almost every brain dysfunction, not everyone, but almost all of them are dysfunctions of inflammation. They're inflammatory based. Where does the inflammation come from? From macromolecules getting through a breach in the blood-brain barrier. That's very, very common. So I'm calling it B4, but that's the mechanism here. Spinach and soy are structural neighbors of human aquaporin 4s of particular interest because this mirrors the dietary habits of the Asian population, particularly Japanese, in whom neuromyelitis optica constitutes as much as half of all of their demyelinating autoimmune diseases. You see the correlation, the possible correlation. You just have to check. You just have to check. When a person has numbness and tingling or peripheral neuralgias, you have to check them for soy, corn, spinach, and tomato. You have to check them because it may be a molecular mimicry going after their own tissue that's causing this. What about wheat and molecular mimicry? Case study number 2, and I've done this one before. We'll do this one kind of quickly, but this is a 3-year-old girl with a hemorrhagic conjunctival lesion in the right eye. Her parents had taken her to a gastroenterologist because of abdominal complaints, and she was diagnosed with celiac disease. She was put on a gluten-free diet immediately. But the gastroenterologist said, there's something wrong with your daughter's eye. We have to get her to an ophthalmologist right away. So they went to an ophthalmologist within a week who found this tumor. Now the little girl's looking down here and they're lifting up her top eyelid. And that's the tumor, big nasty tumor that they diagnosed as Kaposi sarcoma from HIV because mother was HIV positive. But the girl's blood was negative. So they said, what the heck is this? We're going to do a biopsy. So we have to put your daughter under general anesthetic. And they said, no, please. Last week, we were at the gastroenterologist and they put her under general anesthetic and she hasn't quite recovered completely. Can we wait a few days, please? We'll come back, of course, but we want our daughter back to normal before we do this to her again. So they came back a week later. And what did they find? Within a week, look at what happened. Now, this is 2 weeks on a gluten-free diet because they had been on a gluten-free diet for a week by the time they got to the ophthalmologist. And so now 2 weeks later, the ophthalmologist, before they went in to do the biopsy, he just took a look. Well, wait a minute. Let me get that picture of the first time. And thank God they took pictures. And then 2 months later, completely asymptomatic, and the conjunctival lesion did not reoccur 9 months after follow-up on a gluten-free diet. We present a very unusual conjunctival tumor in a patient with celiac disease that showed complete regression by a gluten-free diet. Prompt regression of the lesion during gluten-free diet suggests a relationship to celiac and an autoimmune mechanism, an autoimmune process. There's molecular mimicry once again. Now, a subgroup of celiacs continue to experience symptoms even on a gluten-free diet. I was asked to do a little more about wheat and gluten here, so I'm just going to give you another little pearl. Even minute traces of gliadin or their cross-reactive foods are capable of triggering a state of heightened immune activity in gluten-sensitive people. We've known this for 20 years already. Despite the immense progress in our understanding of celiac disease, little attention has been given to the role that cross-reactive epitopes from various food antigens play in the subgroup of patients with gluten sensitivity and celiac disease whose symptoms do not improve on a gluten-free diet. You always have to look for cross-reactivity. There is significant cross-reactivity with cow's milk, milk chocolate, milk butyrophilins, whey protein, casein, yeast, oats, corn, millet, instant coffee, and rice. Some people have a sensitivity to rice, which keeps them making antibodies to wheat when they're wheat-free. You just have to check. Here's one for you about Candida albicans and molecular mimicry with gliadin, the wheat gliadins. The observation that hyphal wall protein 1. Hyphal wall protein 1 contains sequences of known celiac disease-related T-cell epitopes from gliadins. Candida albicans is present in the intestinal flora of many people. Adherence of the yeast to intestinal epithelial cells is mediated by transglutaminase and a peptide that represents amino acids 40 through 197 of hyphal, whatever that was, which is a tissue transglutaminase substrate. People that have celiac disease symptoms and change to a gluten-free diet may in some cases not respond to the diet. They may be carriers of Candida albicans. They may constantly be exposed to low doses of the gluten-like protein HWP1, thus stimulating the immune system into an anti-gluten reaction. And here is the spectrum of autoimmunity that's got many references of wheat and how it may manifest in autoimmune conditions. Okay, what about lectins and molecular mimicry? Just look at this graph of lectins and molecular mimicry. Lectins from wheat, from soy, from peanut, from lentils, from pea, and from beans. And look at all of the different cross-reactivities that can occur with the nasopharyngeal epithelium, buccal mucosa, the stomach, the parietal cells, the brush border, the colonic mucosa, connective tissue, thyroid, cartilage, liver, pancreas, kidney, prostate. This is why some people do great on a lectin-free diet. And when you do the wheat zoomer, if they come back positive to wheat germ agglutinins, that strongly suggests you want to put them on a lectin-free diet, not just a wheat-free diet. And here are some of the lectin-induced disorders that are well referenced in the medical literature. What about dairy? Well, many of us know that in newborns, especially newborns in a family of type 1 diabetics, you do not give those kids dairy in the first year of life because of the extreme cross-reactivity with the beta cells of the pancreas. So here's beta casein and islet cells, and demonstrating to you the antigenic similarity and where the molecular mimicry will occur. And here it is again with human myelin oligogangliosides and bovine milk butyrophilins. And here are some of the autoimmune conditions that are associated with a cross-reactivity with dairy. What about seafood? Tilapia is among the most common freshwater fish species raised by fish farms, folks, and can cause allergic reactions upon ingestion. It's a common name for nearly 100 different species of cichlid fish. It shows 53% homology to shrimp and 87% homology to human tropomyosins. So we know with shrimp, there's about 100 different proteins in shellfish, and one of them is a tropomyosin that has a very similar relationship, you can see below, with human tropomyosin. Someone sensitive to shrimp, you can now, those antibodies can go after human tropomyosin. What about vaccines? Vaccines are able to elicit the immune system towards an autoimmune reaction. These mechanisms are shared by different conditions. They're triggered by the adjuncts often, leading to what's called the autoimmune inflammatory syndrome induced by adjuncts, or ASIA, the ASIA syndrome. The possibility of an association between hepatitis B vaccine and development of MS was first raised in France, with 35 cases between 1991 and 1997 within 8 weeks of receiving a Hep B vaccine. The neurological manifestations were similar to those observed in multiple sclerosis. Clinically definite multiple sclerosis was diagnosed in half of these patients after a mean follow-up of 3 years. The French pharmacovigilance system responded rapidly to these observations, and they caught several hundred cases with these characteristics. Another example of confirmed autoimmune adverse effects after vaccination is idiopathic thrombocytopenia, which might arise after administration of the measles, mumps, the MMR vaccination. And there are hundreds of studies on autoimmunity from developing from vaccines, not just one. What about environmental exposures? So I showed you about shower heads, but I'm going to show you just one here. And there are, once again, hundreds. You know, there's so many chemicals that we could talk about, just not enough time. I'm going to talk about aluminum, though, because aluminum is used as an adjuvant in vaccines now, very commonly used. And there are a couple of papers presented at the Congress that I just came from, where all of the studies on the safety of aluminum as an adjuvant are based on the dose of aluminum that's in one vaccine, in one shot. And there's not much evidence that that's toxic. But then there are other papers that show that aluminum from the one vaccine has a lifespan of over 7 years, a half-life of over 7 years in the human body. But people don't get one vaccine. They get 6, 10, 12, 22, so that the accumulative amount of aluminum that they're exposed to that doesn't go away for many, many people is where the adjuvant-induced autoimmunity discussions were occurring. So I just wanted to show you this about aluminum. They did experiments with aluminum pans, like you bake a turkey in an aluminum pan, half filled with food solutions and covered with aluminum foil. It was heated to 185 degrees centigrade, which is 365 Fahrenheit. That's the temperature you do turkeys at, right? And the amount of aluminum in the food just kept going up and up and up, depending on the size of the pan and the size of the aluminum foil on top of the turkey. Aluminum is found to leach out from the foil in different stimulants. I really love this sentence, because particularly in distilled water, as well as acidic and alkaline media. What else is there? It's either distilled, it's acidic, or it's alkaline. There's nothing else, right? As far as I know, there's nothing else. But this is what it looked like. This is aluminum foil, the same piece of aluminum foil after baking for an hour and a half at 365 degrees, and the one on the left is magnified even higher. Those pieces are in the food. You can't put your food in aluminum foil to store it. You can't cook or bake with aluminum foil. We have to get away from what we've grown up believing are safe and convenient ways of preparing our foods. I mean, this is a paradigm shift. That's why I said 6 months, because you have to learn about plastic wrap, saran wrap, and Tupperware, and plastic containers, and how that leaches in the food, and how, because it binds to our own tissue, that neoepitope then becomes an antigen that you make antibodies to that activate the entire inflammatory cascade in the development of autoimmune diseases to whatever tissue that environmental substance has bonded to. This is a wake-up call, everyone. So what is predictive autoimmunity? Detection of predictive biomarkers in the early stages of autoimmune disorders can be used to identify, halt, and even reverse autoimmune diseases. Listen to these authors. We sound the warning bell, but we also offer readers the hope of a solution. You guys know these guys are geeks. They're not English majors, but they're trying their best to say, wake up, everyone, wake up. Wait till you read this paper. It's part of the group of papers that you're getting. In recent years, several studies have revealed the importance of early detection of antibodies as predictors for autoimmune diseases. They're markers of future disease in presently healthy people. Look at the date on that. That was 14 years ago that we already knew this. And we've been talking about it. I've been talking about it since 1997 when I did this test when it was research only. And I did a test looking at about 24 different autoantigens. And it came back positive for myelin basic protein, gangliosides, and cerebellum, all in my brain. And I was a healthy 44-year-old guy. I was doing great. And I said, this is a mistake. I called the lab. They said, no, it's not. I said, do it again. They said, we did. We know it's you. We did it again. Sorry. And that's when I woke up to this. And I think every one of you needs to do a predictive antibody panel to see what you're personally dealing with right now. Unless you want to wait. You wait 20 years, 25 years, and now you've got cognitive decline and early dementia. And then you have to follow Dale Bredesen's protocol and hopefully get some results. But it's much harder when you kill off more tissue. Everyone needs to do predictive antibody panels on themselves, because then you look in your patient's eye. You say, no, I did this on myself. I can't believe I had antibodies to my joints and my muscles and my eye, and I had a little blurry vision. And I'm fine now because I fixed it, Mrs. Patient, and we're going to do our best to help you too. But if you don't do it yourself and you're just peddling this because it's a good idea, you don't have that same gut feeling that nobody can argue with you on because you've experienced it for yourself. So this is the first panel I do for almost every patient that we see at thedoctor.com. We look at total immunoglobulins first to make sure that this test is going to be valid for them. And then I look for indicators of intestinal permeability, because this talk today is not about this, but I think most of you know intestinal permeability is the gateway into the development of autoimmune diseases. You've got to fix the leaky gut. You've got to. So I look to this first with practically every patient that comes in. And then here's the neural zoomer. They have any neurological symptoms whatsoever, anything. Little numbness and tingling once in a while, little brain fog once in a while, we do this panel because it wakes people up and it gives them the window of opportunity to do something when they have a problem. By the way, I recommend the ApoE4 to every patient, or the ApoE test, because if they have an ApoE4, you sit down with them, you spend a little extra time, you look them in the eye, and you say, we've got to have a serious discussion here. And then you tell them the statistics on Alzheimer's and ApoE4, and then you say, but every one of those studies, Mrs. Patient, every one of those studies were people that kept living their lifestyle in the pond of brain dysfunction. Whatever their lifestyle was, the foods they were eating, the bacteria they were exposed to. And then you tell them about Dale Bredesen's book, The End of Alzheimer's and Reversing Alzheimer's, that we can help with this. And you're an ApoE4, Mrs. Patient. So it's really important that we address this. If you think it's important, we're happy to address this for you and give you our best in stabilizing this. That's how you use that test. And Vibrant will be coming out, and it's a little premature. I mean, I had a talk with them a little bit ago, and so they're revising the panels that they were going to come out with. So I just want to show you the panel that they were thinking about for rheumatic-related diseases, like the arthritides and systemic sclerosis and lupus. So here is one panel that they were putting together that's being modified now a little bit more. This is just page 1. All of these antibodies, all of these different antibodies are being looked at in one panel for musculoskeletal complaints. I mean, this is just fabulous. What's coming down the pike is going to be so fabulous for us. And you guys, if you take the next 6 months and really dial down on the papers in this book, when these tests come out, which will be sooner than 6 months, but when they come out, you'll be ready to go. You'll be ready to go with this. Individuals who are at risk of developing an autoimmune disease should be advised to refrain from activities and lifestyle which endangers their health and quality of life. And, you know, I'm in Chicago right now. I just came back from Portugal, and I'm in Chicago. And the way a Chicagoan would say that is, you think? Of course. But we have to know how to counsel them. And to know how to counsel them, you have to do the right functional test to see where the problems are. You have to go back upstream, get out of the water of the pond, walk up the hill, walk back up the river to figure out what fell in the river that carried you downstream and you crashed over the waterfall, Mrs. Patient, into the pond below. And here's what predictive autoimmunity can do for you. PPV stands for positive predictive value. If you have antibodies elevated to lupus, it's 94 to 100%. You're getting lupus in 7 to 10 years, unless you change something. Rheumatoid is 52 to 97% in 14 years. Primary antiphospholipid syndrome, 100% in 11 years. Hashimoto's, especially postpartum, 92% in 7 to 10 years. You're getting Hashimoto's if you have elevated TPO antibodies. Primary biliary cirrhosis, 95%, 25 years. Crohn's, look at the Crohn's, 100%. If you have antibodies to Saccharomyces cerevisiae, which, by the way, they have homology with Saccharomyces boulardii. So if you're supplementing with Saccharomyces boulardii, you better make sure they don't have Saccharomyces cerevisiae antibodies, because it's 100% positive predictive value in 3 years. They're getting Crohn's. The importance of screening healthy individuals lies not only in the ability to prevent life-threatening manifestations such as Addison's and thyroid storm, but also in the ability to treat and even prevent overt autoimmune diseases. I just couldn't resist when I saw this one. You know, this has been an intense hour, I know, but you know, to bring a little humor into it. So for those of you that haven't seen Betrayal, that was Dr. Terry Walls was in Betrayal. That was a clip from there. We interviewed 85 of the world experts. All this is free online. We've had over 500,000 people watch it. I'd recommend that you take a look at this at thedoctor.com/betrayal, and then recommend it to your patients, because they then come, and there's nothing being sold, no nutrition, no tests. We talk about nutrition, we talk about tests, but they're to go back to you for the treatment protocols. But they'll come back to you, and some doctors even play this in their waiting room just on a continual reel. And the result is patients are asking really intelligent questions when they listen to Betrayal. And for those of you that really want to dial down gluten, I have an online certification program and I'll just say one thing about that, and that is in 1999, the Journal of the American Medical Association, 2009, in JAMA, the largest study ever done on mortality in celiac disease. They looked at 39,000 celiacs. What they found, one of the little points, it's only 2 lines in the whole paper, but it's so critically important. What they found was that adults diagnosed with celiac disease have an 86% increased risk of mortality in the first year after diagnosis from cardiovascular disease, after diagnosis, compared to someone not diagnosed with celiac disease. That's it. 86% increased risk of death from a cardiovascular incident in the first year after diagnosis. What's different for a patient after a diagnosis of celiac disease? You put them on a gluten-free diet. What else? Nothing. And the result, 86% increased risk of mortality. So that's the kind of information you get from the CGP program. Why does that happen? What do I do about it? And all of that.